Or no, you guys are in it this. Okay. Yep.
Last semester.
All right, we're going to get started.
Oh, and then
And I think we might have maybe a couple new people. I have a couple more physical copies of syllabi if you need one. And if you lose this, anybody loses them, it's also on a Brightspace. And so we've got a Brightspace page, but I will kind of almost never use it. I talked about that last time. Also, if you weren't here last time, I record these lectures, so you can totally catch up. And so if you weren't here on Tuesday, send me an email so I can send you that recording link. I sent that link out to everybody else. Then we talked through the syllabus, so really soon watch that, because we talked through the syllabus and there's a requirement that if you weren't in class, you have to take notes and send it to me, because you have to participate. I'm recording it for a very specific reason, and that is I want you to get a chance to learn the stuff like that. So we're gonna, everybody is gonna be able to catch up and be right on track with that. And so we take 10 minutes like that. If your name's not on here, just add it. I am sending things out. I'll email out, a couple of emails a week, like with the links to our recordings, and I'm using your USM email addresses. So. And I'm also using the list of registered students, right? So if you're not registered quite yet, also let me know that so I can keep you in the loop like that, okay? Yes, and so welcome, and then we'll start with this. What are we doing this week? So in those early emails, but also posted on our Brightspace, are these links to about a half hour worth of videos that I made that we're watching. It's a first priority, and we're watching them, and they're about muscle differentiation. So in our bodies, make new muscles, and we have to make new muscle cells. So it's about that. It's used that as example, but we'll talk about it today, how it's a universal example for what we're really interested in, and that's the organization of genes and gene expression, the output of gene expression. And so we're really focused on those. And at the end of class on Tuesday, we're going to have a quiz. So you can bring questions about them today, or even next Tuesday we'll have a quiz. At the end of class on Thursday, we're going to have an activity, which is really just physically working with clay and organizing and understanding the physical structure of genes, like that. Okay? And so that's our top priority. As you're watching them, get in the habit of writing down questions, whether they're I'm just confused questions or specific questions. Joe sent me a very specific question, and we'll cover that, like that, okay, in class, like that. Sometimes I'll just email your answer. Sometimes I'll be like, okay, when I have that in class. So you can send me questions, but it's really great to bring them to class, like that. So we'll have time to do that. And then we've got, you're in the wrong classroom. Yeah, okay. Yeah, it was going to be hard. Yeah, okay, cool. Yep, okay. Good luck. Yep, and that was kind of always my nightmare in college, right? Only in my case, I'd be like halfway through the semester, and I'd be like, oh, whoops, let's see. And I talked to a student yesterday afternoon, Mike, and Mike had slept through the semester till then. He just had gotten up, and whenever it was Wednesday, and he's like, oh. Like that, and I'm just like, good try. So even if you're in class, you might want to watch the class recordings again, or parts of them, right? You might space out, or you might be confused, and you might realize that you just want to drag back through it. One of the things is I'm going to tend to delete them after a week. I don't want you to think that the night before exam, you'll just be able to watch them all and learn them all then. I want, it's got to be ongoing learning. So my plan is to delete them, but give you a chance, like a week, like that, okay? And because even if you're in class, there's a good chance, I want to watch them again. And then the next thing is we're going to start reading chapter 1, which is an overview, and we're going to go chapter 1, chapter 4. The Brightspace has a link to those two chapters. The first chapter is going to read 1 and 4. So even if you don't have our book quite yet, you can start looking at them and write down questions, bring them to class. I talked last time about that, and the importance of bringing questions to class. Another thing I talked about last time is you know, when I was a kid growing up, I was out in nature, and that when I went to college, I wanted to be a scientist because I really liked animals, and I liked that kind of level. And then when I was in college, I ran into this molecular biology, so nuts and bolts stuff, and it just hit me like a lightning bolt when I was exposed to the topics in this course, and I, you know, stole the book. It was years later when I actually realized, oh, that was evidence of a passion. You know, everybody says, oh, follow your passion, right? Even though I ran into it like that, I didn't even recognize it. Oh, that's my passion, you know, like that. Like it can be really hard to say, oh, I think I found my passion. And I like to tell students now, if you're willing to like break the law to do something like steal, or, you know, that's probably something you're passionate about. So you might want to go to jail for that and then have a career. The other thing I want to say, it was also years after that I realized what was going on. And that is, I grew up in a little farming town. I loved nature, but I also worked on farms with tractors and implements, which are the big pieces of equipment tractors pull. And I really liked that. Mechanical nuts and bolts, like car mechanic stuff, like, you know, just a fact. And that's what this is. So I talk that way. You know, I talk like, you know, boom, 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 like that. I also want to emphasize that I think molecular biologists are also the most brutally skeptical scientists. So even though we'll be talking about that's the way it is, it'd be great, you know, if we discovered something that changed those truths like that and updated them. That's great, right? And that's happening. A lot of this stuff is just really, really well established. And it becomes a thing where we're very skeptical that it could not be, you know, the way things are. You know, I'm skeptical of that. But man, super skeptical. And that's because molecular biologists have the tools and the setup to understand things really carefully, right? In lab, controlled experiments, and just little pieces, trying to, you know, add a little brick of fact to the big wall of facts, like that, right? And so molecular biologists can just sit back and say, I see that you proved it that way, but I'm not convinced. Prove it in an independent, different way, like that. So I'm excited like that. I'm talking like that. This is kind of a car mechanics course, okay? Like that. Okay. So then we'll stop today about our way through, and we'll do one of these concept chats where you'll look down at your notes and you'll say, what are the 3 important things that you think we talked about? And you'll chat with a partner about that, you know, just randomly chat with people, and then I'll ask, you know, what did you think was important today? One thing important today, you know, like that. So we're trying to get that going to help you be ready to ask questions like that. So we'll do some of that today, and that's that. Then there's this 4-H STEM Ambassador program, and I said that's an option to do either a term paper, which will be graded on, or this, which if you do, you'll just get 100. So I want to encourage you to do that. And Godwin, did you do this last semester?
Yes.
Godwin did it last semester. You can do it again, like that. Yep.
Okay. I just wanted to know, like, how much of a time commitment that is.
Good question. Did anybody else already do that 4-H STEM Ambassador before in some setting? Okay. Because I think if you did, Godwin, I don't think you have to do the training, or maybe you just do one.
So, it's pretty easy. It's not
Yeah. And so you could also ask Godwin, you know, what's the expectation like, okay? There's an organizer. I don't organize it. There's someone named Sarah Sparks. The link to the website and the registration and the schedule is on our syllabus. And so you go to Brightspace, find our syllabus, and just click. You know, you don't have to copy it down, just click on it. And on there also is Sarah Sparks' email address, like that. And so if you have questions like that, just let her know. Next thing is, on that website is this training, and students almost immediately said, whoops, we have a lab in the afternoon, okay? So you do 3 training sessions, and there are options. So 3 1.5-hour training sessions. So the first thing to say is time commitment, 4.5 hours of training. It looks like these first 2 are Zoom type settings, and then the third one is an option of Zoom or in person. It looks like that. Then people said, oh, we have a conflict. But you see, like, yeah, they're on Tuesdays, but one is later in the evening. And even one, it's 4. And if we could Zoom, and if there are a bunch of people in a lab who want to do it, we'll just have them Zoom right in lab. You can just do it in lab, right? Start at 4, you know, because our class is supposed to go to 4:20, and we'll just make sure. Because I want to be supportive of this, right? But, you know, everybody, you know, could likely do this, because although it's on Tuesdays, Sarah set up some different times, and we can talk about it like that. But I super encourage you to do it. You don't have to tell me you're doing it. You would tell Sarah, and you would apply and go down that pathway. So first thing, time commitment. 4.5 hours of training, and then I believe it's 4 one-hour class visits. So 8.5 hours of actual time. But for the 4 class visits, you're going to be given a kit of a STEM topic, and you're going to teach it. And like I said, it probably won't be biology, but be like wind power, or there's one about some fish called alewives, which is a biology topic, and one that's a little bit about computer type stuff. You know, it's a STEM topic, and you just have to prepare that. So it's 8.5 hours of training or class time, and then some time to prep. And you'll have a partner, so you'll communicate with them, and you know, that prep time. And that would be, I don't know, Godwin says it's pretty easy, like that. So we can talk about it. The thing is, you know, you have to decide by then, right, coming up, if you're going to do it or not. The term paper is still not late in the semester, but this is something we get going on like that, okay? So there's that. Then Riley. Riley's going to be a tutor. He's coming here a week from today to start teaching us about CAR T-cell therapy. And we're going to think of him as a novel person who's a tutor, but also an expert in this CAR T-cell therapy, which is a type of cancer therapy that involves a couple of key words that we'll learn something about. One is synthetic biology. So we understand the organization of DNA and how it relates to proteins so well that we can manipulate that in a test tube. And I even talk about that a little bit in the clay model videos, and we'll talk about it a lot. And so the CAR T-cell therapy is a type of synthetic biology where they build a cellular receptor for the patient's immune cells, and they build it by manipulating the DNA in a test tube, and then they insert it into the patient's cells. So it's one thing. Then it's in cells. So it's also another term that's emerging, and we talked a little bit about it. It's called cellular therapy. The tool is not a medicine or a surgery or radiation. The tool is actually a modified cell, and you're harnessing the power of that immune system cell to attack the cancer. So it's good stuff. Riley will be here a week from Thursday, and he took this last year. He's a student here. He's great, like that, okay? There's a little thing of that. I mentioned that. So then here's something I didn't mention last time. I help organize an internship, a research internship, and it's a Maine Health Institute for Research in Scarborough, and it happens each school year. So it goes from September through May. And so this is if you're going to be around here next school year. It's fantastic. It's just fantastic. And Eva is doing it right now, and there are about a dozen USM students that are down there right now. So you could ask Eva about it. And Eva's having a really good experience there because her PI can speak German like that, which is a highlight, right? And a surprise because her PI is from Russia, Igor Prodovsky. But Igor is just a freaking genius and can speak probably any language like that. You know, just like, oh yeah, I know that. Yeah, it's a really good experience. It's this kind of stuff. It's at a medical center institute, but it's really using mice as a molecular genetic model for human health. And so they study things like blood vessels and circulation, hearts, fat. There's different kinds of fat cells in our body doing different things. It's very, very big deal in biology right now, medicine right now. Some cancer biology and stem cell biology and a lot of this kind of synthetic biology type stuff, understanding it, using mice as a molecular genetic model like that. And so I'm the contact person at USM. If you're curious about that or interested, the applications for this next school year will come up in March. And so, let me know if you're curious or interested about that, because it's a fantastic opportunity. And of course, there are others, but this is one that's just off the charts in its quality. And it's a paid internship and so on and so forth, like that. Okay, good. Here's just a picture of one of the lab groups, Michaela Reagan. And Michaela works on multiple myeloma, which is a type of blood cancer, and a couple of USM students that were there several years ago, like that, okay? So, on to our course. So let me pause and check in if people have questions about anything at all, whether it's about the course or about the videos or anything. One thing that will come up should come up is that the cellular and molecular biology has exploded into every topic in biology. And so you've learned some of this stuff. I mentioned that the first semester intro biology is always a cell and molecular biology course. So you've learned an intro of it, but also in your other courses, whether it's genetics or ornithology or just about anything, these things come up like that. And so you know some of this now, and one of the things that's really important is if there's something that seems contradictory to something you've learned, you just bring that up. And you don't have to be certain about it, and you can even misstate it, But if you say, oh, we learned something about this in, A&P, and then you try to say it like that, that's great, right? That's just great because you're learning and you're, and it might have been like not perfectly described, or you might have absorbed it not perfectly like that. So you have what's called a misconception. And we would like to address that and talk through it like that. It's really good. So when that comes up, write it down or in class, just say it out loud, okay? So that's where we're at. So we're gonna start on talking some about this, okay? All right, so what's going on here is a timeline, right? Early development, and then we're gonna form these muscles. So we're on a timeline, and we're just picking a short segment on the timeline right before the muscles form. So this is a muscle precursor cell, and then a little bit later, it's a muscle cell. So that's our example. We have 2 cells. And then over the time, there's changing gene expression. And we have, on this timeline, 3 genes. 1, 2, 3. The first gene is myogenin. It gets activated. Myogenin makes a protein that's a transcription factor that binds to the second gene, myogenin, and activates it. Myogenin gets expressed, and it's a transcription factor that binds to the third gene, and those are muscle genes, like that. So we're on this little timeline. And you might say, what happens earlier in the timeline? And in the timeline, we're just focused on that part. You can ask about it, but we're just focused on that 1, 2, 3. And in that, we're really focused on the myogenin gene. That's the clay here. So we really have 3 things that we're talking about. We're talking about a particular set of cells, the muscle cells. Then we're talking about a particular gene. We're interested in the myogenin gene. And then we're really focused on this MyoD protein. 3 things. Okay? This example is actually a universal example. So let's just start with that idea. So 2 cells, and probably the most common experiment in all of biology is you want to compare 2 cells. And sometimes it's like this, a myoblast and a muscle cell. And you want to compare them in an experiment and see how they're different, right? How are they similar, how are they different? But lots of times, right? Here's a cell that is a muscle cell, versus a brain cell. You want to compare muscle and brain. Or here's a cell that is healthy, here's a cell that's cancerous. Or here's a young cell, there's an old cell. Or here's a cell with no medicine, there's a cell that had medicine. So you can think of it like cell A and cell B. And so that's the first thing, is that we're probably talking about a cell example in our case, but it's universal. We really want to do it. Now, sometimes it's a timeline. It's not just early and late. It's like, time 0, 1 hour, 2 hour, 4 hour. Or it's a concentration. No hormone, 1 millimolar, 2 millimolar. So sometimes it's 4 cells or 5 cells, but you're really comparing those cells. Okay, so that's the cell thing. What's the cells we're talking about? Like that. Okay, the next thing is then we learned about this and are learning about this concept of genomic equivalence. The cells always have all the DNA. They have the whole genome. One of the questions on the quiz is going to be short answer questions. It'll be like, what's a genomic equivalence mean? And you say, with rare exceptions, every cell has all the DNA, has the whole genome, right? And that's just startling, right? All our cells have the insulin gene. All our cells have the myogenin gene all the time. We can get, the DNA can get methylated. We bring that up, and we'll talk about that. But the letters A, G, C, T, those really aren't changing. So the next thing is we want to talk about the DNA, and the DNA is not changing. We could get mutations, you know, so sometimes, like say in that cancer example, there's probably some mutations, so there is a difference in the DNA that they're really, you know, trying to figure out too. But in general, you know, we're pretty much born with the same DNA that we die with, like that. You know, we get some mutations, but really that's not changing. Yep.
Were you just saying that all cells have the same like genes? Yes, but also like components, or was how did you phrase that? That they
Just
all have the same-
all the same DNA,
Okay.
like that. And that means like that letter code, AGCT code, that's just not changing, right? And DNA is a information storage molecule, so we're saying we got the same DNA that we inherit from our parents for our life. And that's true for everything on our planet, with some rare exceptions. There are some immune system cells in our body that rearrange some of the genes like that. Super rare and unusual, you know? And that happens in an organism here or there where they actually alter the DNA a little bit, you know, like that. But in general, plants and animals on our planet, always the same, like that. And so the DNA is the same. So the next thing is we'll just draw on some DNA, and you know, we can say, you know, that's going to be like this, and that's the DNA, and it's not changing. And we can study the DNA, and we do it using an instrument called a DNA sequencer, and we sequence the DNA, and we read the letters, and we got it, like that. We have that information. We're set. But we don't need to do that when we're comparing these two cells, because they're going to have the same DNA. Now, maybe they have a little DNA methylation. We can talk about that, but the letters are just not changing. So we wouldn't study the DNA in this story. We'd already know some about it, like that, the DNA sequencer. What we're really studying a lot is this output, right? Because that's a key thing. What about the output? Do these cells have the same output or a different output? Has that changed like that? And it has changed, right? So this would be RNA. That's the output. And I've never read somebody teaching it this way, but it's just the way it is. DNA is an information storage molecule like the files on a computer. And when you want to output it, when you want to open them, that's what that gene expression is like, is you're just opening it, right? And when you do that with a computer, you just open the files you want. So when we copy this, we transcribe the DNA into RNA. That's gene expression. And here's the word gene, because we don't open all the information at once. We open like a portion of it. And that portion is a gene. A gene is like one file in a computer, and you just open the one you want. Now, that can change, right? From comparing these two cells, that can change. So an experiment that's just super common is measuring the RNA levels. For myogenin, we would see that myogenin gene, no RNA in the myoblast precursor, and then has RNA in the muscle cell, right? The DNA is not changing, but the RNA sure is. And so a really common method, right, in comparing cells is to measure differences in RNA. What genes are being expressed? The brain and muscle, they have the same genes. Well, which ones are they using? Which ones are being expressed, right? Because that really explains the story of the differences between them. And so there are methods that scientists use to measure RNA. And they include things like PCR, and a variant of that that's called qPCR, and then something called RNA-seq. And we'll learn about these because scientists use them a lot. And then an older method that's called DNA microarray. And even an older method that's called a northern blot. And those are all methods that are desperately interested in measuring the RNA levels. And we could do that for the myogenin, and we'd find that's our result. No RNA here, lots of RNA there. Sometimes it's just like a yes-no thing, like that would be. Sometimes you want to quantify it, how much. You know, maybe there's some and it's increasing, right, like that. That's super common.
And you use those to sequence the RNA or read
So,
the RNA?
yeah, so, or detect its presence, really, is that's what you want to do, is detect if it's there, okay? So we're super interested in that, and we're also interested in the relationship between the DNA and the RNA. So I'm just going to redraw this, because it's getting messy, and say, in these two cells, the DNA is the same, and then, how's the DNA organized? And it's going to be the same, and we have this portion of the gene that is going to get transcribed into RNA, like that. And it's getting transcribed here, so that's transcription, and that's being done by RNA polymerase, the enzyme RNA polymerase. And it's not happening here. So we can detect this RNA. And that DNA, that part of the DNA, is called the transcription unit. And then scientists immediately are interested in, well, how do we know that that should be activated? And the answer is all the genes have two parts. One part is the transcription unit, and then the second part is next to it, and it's like the regulatory region, And see, I'm putting those in color because those are made of DNA, and that's another part of our quiz, is that. And it's described in the video, so I'm breezing through this. But you'll see we're going to add some stuff to it. So there's the regulatory region, like that, okay? So we have some structure of DNA that explains how we're getting this output. Now, we said, well, the DNA is not changing. The DNA is static. But we're describing something that's dynamic. There's a change here. So what is really happening? What's the difference here? And the answer really often is that there is a transcription factor, which is a protein. And sometimes it's more than one that's not active here, and it's active here. And that's what's making this happen. And so really often, in comparing these, the dynamic story is that. That's like the switch. The DNA is static, and the DNA is just a landing site for this switch protein. And it's a protein, and in humans, there are about 2,000 transcription factors. And we've already learned two. One is MyoD, and another is myogenin. Okay?
What activates a transcription factor?
Yeah, so that's a good question, like that, because it's like, how does it get switched on and off? In our first example for MyoD, the story we picked is kind of simple because it's just, MyoD gets made, and it does its job. So that's the switch aspect of that. And I'll just show you this now, since it came up. And that is, you might have to scroll a little bit for this, don't worry. Yeah, here it is. Okay? So one of the big things we'll talk about through the course is transcription factors, and that dynamic story of transcription regulation is, well, how are those being regulated? So how are you switching it? So then we're gonna, over the semester, build a list of probably a dozen examples of how we could switch them. So your question, and the first one is, well, it's just that the transcription factor needs to get synthesized. And our example is MyoD during muscle differentiation. The MyoD is not there, and then it gets made and it's active, okay?
And that activates the transcription.
Yep, that's the switch. Now you might say, well, what's upstream of that? And remember I already said, well, we're just gonna talk about a segment here, because the story that what activates MyoD is a good question, but it gets complicated. We have a nice crisp one to start our course with, and that is, MyoD just needs to get made. And when it's there, it binds to myogenin and it activates myogenin. One way to think about that is anytime we can detect myogenin RNA in an experiment, we could also detect MyoD protein, because they go hand in hand. MyoD protein gets made and activates expression of myogenin. Okay? let me just show you this since we're on the topic. Last time, we jumped way ahead in our course to chapter 17, which we're going to get to pretty quickly, and we say, oh, look at this FOXO. FOXO is another transcription factor. And look how it's regulated, because it's a nice switch, right? It's being excluded from the nucleus. So that's already our second example. The transcription factor can't get to the DNA, right? The DNA is in the nucleus, and it can't get there. So our second example is it's excluded from the nucleus. So that's one way to regulate gene expression. So in some examples, that's what's going on. Oh, this gene isn't activated because the necessary transcription factor can't get to it. Then in this one, we already even have our third one, and that is another part of the FOXO story is that it gets phosphorylated. And phosphorylation is a switch, on or off. In this case, phosphorylation prevents it from acting. So that's already our third thing is, well, you could be phosphorylated by an enzyme called a kinase. And we'll talk about that more because we'll loop back over that again and again. But we're making a little list like that. And we'll make it over time. And you know, sometimes if you're like, dang it, I didn't get all that written down. Remember, we're recording this, right? So that, you know, you can get it. And I'll try to, you know, not go ahead, right? But just to, you know, really kind of address your question and kind of move forward to see, oh, we're going to have some different examples. And that's actually one of the big picture items from our course is transcription factors are proteins. They're super interesting because they're dynamic. They're switched on and off. How's that happen? Like that, yep. Okay? So then, back to this, right? So now, that's our story. We have some cells, we have DNA, and a specific gene, and then a lot of times there's a transcription factor story. And in an advanced course, there's more stuff added, but we'll kind of beat this a lot. And in a sense, you could do this where you would draw this drawing, and if you left the labels out, you would just have blanks like that. And the blanks would be, you'd say, well, what cells are we talking about? And then the next thing is, well, what gene are we talking about? And then the next thing is, well, what transcription factor are we talking about? So in our videos, we've got a specific example, but then it's also a general example. And that's how you would slide it around. It'd be the same drawing, but you just switch those labels as an introduction. Yep?
For a promoter, is it in the regulatory region? Or
Yes.
description?
So that's that. And so that's the first thing I want to say is this is a universal example. Okay? Then the next thing is we really want to zoom in on that DNA and the gene organization like that, because that's also universal, right? And there's a thing about molecular biology where I think, like, I don't want to criticize any other professors, but right, molecular biologists are just like like that, you know. We want to fix this tractor, so we're going to figure out the parts like that. So it's like that. It's just like, are these facts? Yeah, they're just like that. Boom, boom, boom. Really makes sense to make flashcards and flashcards that lay out on the drawing, you know, because it's that kind of knowledge. And there are many examples, right? Those 2,000 transcription vectors in us, at least half of them have been studied well, you know. So we're going to learn a dozen examples. We're really picking and choosing, but man, oh man, some of those examples are so well studied like that, okay? And that's not intended to try to overwhelm you. It's just trying to just say that when we say, you know, look at this, look at this, look at this, there's a lot of evidence to make us want to summarize it and introduce it this way. So now we're going to look at a gene and that circular gene. And so we're just going to draw that green line out, and it's DNA. So here's some DNA. Struggling a little bit here. And we've got this universal organization of genes. 2 parts, and they're physical. Oh, my little pen is getting jumpy here for some reason. I'm too enthusiastic. I'm just going to sit down and work this out. So one part of it is the transcription unit. And our example is myogenin, like that. We'll get to that. But right now we're just talking in general. And then we have this regulatory region, like that. The regulatory region regulates three different things: where, when, and how much of the gene is going to get transcribed. And you can just memorize that because that's just the way it is. Where, when, and how much, like that. And the next thing to say that is a surprise to hear is evolution involves random mutations and the formation of new alleles, randomly forming new alleles that might be beneficial, like that. And those mutations are really typically in a gene, and there are only 4 ways to mutate a gene, to alter it in evolution. 4 ways. And one of them is this. The mutation could be here in the transcription unit. This pen is just kind of not perfect here for some reason. So you could have a mutation in anywhere in that region, and that's going to affect the product of the gene. So we could have mutation that affect the output product. An example of that is like sickle cell anemia. People who have sickle cell anemia have hemoglobin that is altered and is sticky. That's a story about a problem with the product. And we know for sure the mutation must be in the transcription unit, right? That's one of those tractor farm town logics. The product is defective. The mutation's in the DNA. What part of the DNA? It's the transcription unit. The other 4 would be up here in the regulatory region, and they could affect where, when, and how much. And that's it. It's impossible to have any other category of mutation in a gene. So that helps you see that, oh, the regulatory region is actually really important. And of course it is, but you can see there are a variety of mutations. And there's been a lot of evolution, not by altering the product, but altering where, when, and how much it's expressed, which, of course, is particularly important to talk about genomic equivalence. The genes are there, and we could have a mutation that changes the gene's activity. The gene is there, so we could all of a sudden express a gene in a new place because the DNA is there, and we might just have a mutation and say, where are we going to express it? It's a new place. And you're like, that's probably bad, but it might be good, right? So that's that, just to talk about that, okay? Now, let's just stick with mutations for a little bit, okay? So let's, I'm just going to redraw this same thing, but just kind of move it over a little bit, I guess. And we're going to stick with mutations a little bit and say, now let's specifically talk for a little bit about myogenin and these mutations. So myogenin, and it's the gene, and it's going to make a transcription factor that's going to activate muscles. And it's going to be activated by MyoD. Okay, so we could have a mutation, and the mutation could be in the transcription unit, and that could cause us to make a defective myogenin protein, and the phenotype would be no muscles. And people call that the mutant phenotype. If you don't have myogenin, you're just not going to get muscles. We could also have a mutation up here in the regulatory region. And let's say it's the part of the regulatory region where that MyoD has to bind. That's called the enhancer. It's part of the regulatory region, and we'll talk more about it. But there could be a mutation there that would block the binding And in that case, we have a nice myogenin transcription unit. We could make a nice protein, but we don't make it because MyoD can't bind. Like that.
So
Okay.
where, when, how much is effective?
Right. So there's a mutation for how much, and it's like none. Can't make any, right? So now let's look at this, because that you can have a mutation in the transcription unit or in the enhancer, and they could have the same phenotype, no muscle. Then let's look at this. Well, that MyoD is a protein, so it must have a gene. Here's the MyoD gene. And there could be a mutation in it, and it could be in the transcription unit, and we could make a mutant MyoD protein that would be defective. Now in this case, now we have a normal wild type myogenin gene. It's normal. But the mutation's way over here, which is typically like a different chromosome, far, far away, but yet same phenotype, no muscle. Like that. So geneticists studied these kind of things, and they were really curious because they're like, in the genetics of it, they see, oh, look, we have these mutations here. They give us the same phenotype. And then we have this mutation over here that's a different phenotype. So in genetics, there's a term. And the term is that these mutations that could occur on this gene would be called cis mutations. And what that means is they're clustered and they're physically connected. Then this mutation is way over in a different gene, and you know, geneticists could map that out, and they call that a transmutation. And you know, back in the day when you just had genetics, no one was really curious what's going on. And so they use that term a lot, and they don't so much anymore because it's kind of obvious when you know the DNA sequence and the organization of the genes. But that's
So-
an old genetics term. Yeah.
So you could have differences in the gene mutation, but the output is more or less the same.
Yeah, in this case, exactly the same phenotype. No muscles, no muscles, no muscles. Like
Well,
that.
you said the phenotypes were different because they were in different locations.
That's not the phenotype, that would be the genotype. They do have different mutations at different locations, but if you studied the organism, like a geneticist in the 1950s, you'd say, wow, each of these mutations that we have mapped to different chromosomes have the same phenotype. Even though some are close together on a chromosome, same chromosome, and then some are on that other chromosome. So they invented a term to describe that pattern of mutations, and the term is just cis, means they're all connected, okay? And trans means they're in another place. And you can see that, you know, with genetics, you can provide evidence for our clay model video. You know, that clay model video is a combination of genetics evidence and biochemical evidence, like that. Yeah, question?
For the cis and the trans, you're saying, is it like just on the same chromosome for the cis, or do they have to be like gene regions that are just like really close, or like beside each other, or like?
Yeah, so the best thing to say is that cis means physically connected.
Where? How?
Yeah, by the DNA, the strand of DNA. And in Joe's question from the videos was, in the videos I say that, this region right here, that DNA right there, that DNA is like a tether or a leash. And what does that mean? So what we're connecting in this is, it's the DNA, but it doesn't matter what the DNA letters are. It's just that it's like a string, a piece of rope, like a leash. And what we're leashing or tethering is this enhancer is tethered to the start of the transcription unit. Because what we're doing with this region is we are leashing the MyoD protein. So it's close here. The DNA is floppy like a leash. And so it's flopping around, and what we've done is we've forced the MyoD to stay here. And MyoD's job is to do something here. That's what it's supposed to do. And so that's what I mean by a tether. And scientists showed that because when they discovered enhancers, they discovered that you could move the enhancer as long as it stayed physically connected by the DNA. You could move the enhancer, so let's just make a special color for enhancer. So you could move the enhancer experimentally. You could move it further away, and things would still work. You could move it closer in, it still work. This like a leash, a little bit longer, a little bit shorter, doesn't really matter. You can even put it down here, and it still works. The way to think about it is like imagine that MyoD is like a bitey dog, and it's running around your neighborhood because the MyoD is just floating around in the cell. There's always lots of it, but it's floating around in the cell, and it's just not going to bind to the transcription unit and activate transcription. And we'll learn later in our course, well, how does it do that? How does it affect the RNA polymerase? How does it increase the rate of transcription? All right? But you start out with that. It's just running around the neighborhood. Now, you put the dog on a leash, and the dog's running around, but now it's running around in a very small area, and you're going to get bit, right? Pretty soon. The first time, it's a football field, and you're like, I hope I don't get bit. I probably won't. But now you're going to get bit, and that's exactly what's happening. So scientists like to use, and it's kind of an insider term, but they talk about local molar concentration. You kind of have increased the molarity of the MyoD in that region, in the way you would do with the dog, right? You didn't really increase the molarity, you didn't increase the number of dogs, but you just decreased the volume that that dog is active in.
And
so
that's just by moving the enhancer?
No, it's just by having it tethered.
Okay.
That tether. So like when we circle this, we say that's the regulatory region. The regulatory region needs to be a physical stretch of DNA, but for most of it, the letters don't matter. It's just a leash. And there are just really two regions that matter. And one is the enhancer, and that's really where the big transcription factors that really regulate things like MyoD bind. And they're just caught. And they're cis. That's what cis means, is there's something about how that enhancer DNA needs to be cis or physically connected to the transcription unit, right?
So the regulatory region, which is the area between the enhancer and the transcription unit, is just physically the leash, and having that leash is what activates.
Yeah, so let me just erase this and show you that no, it's more than just that. So what it is really is the regulatory region is really basically two places. And so here's the transcription unit, and then here's the regulatory region. And we go like, oh, it's here, like that, And that's because of that tethering thing, because it could be a big region. But there are really two regions that matter, and one is right here, and it's right next to the started transcription unit, which is referred to as +1, because +1 is where the RNA polymerase starts, and it starts copying, transcribing, and the first nucleotide is 1, and then 2 and 3, so people just call it +1. And this region, we call the promoter, and we'll get back to the names in a second. And the promoter's DNA, and there's some transcription factors that bind here. And their job is really to show the RNA polymerase where to start. Then there's going to be this enhancer region, and that's can be far away. And it's going to bind an important transcription factor like MyoD and just keep it in this region. The promoter transcription factors are generally kind of boring. The people who study them, of course, they're not really boring, but it's more advanced to talk about how they're interesting. For us, the promoter binding proteins are kind of boring. It's the enhancer binding proteins that are the big switches, that the ones that we're going to make a list of, like MyoD, myogenin, FOXO, like that. Those are the ones, right? And so that's the regulatory region. And it's like this thing where you go, it's got specific regions, and then it's just got the stretch of DNA that sequences don't matter, like that, okay? Like that. And just to put numbers on them, this enhancer is really typically about 20 nucleotides long, 20 letters long, and the promoter too is like, say, 30 nucleotides long. So really short stretches. And this overall regulatory region could easily be 10,000 nucleotides, because it's really just serving as the leash to tether it, like that. So the question was about that. Now, the next thing that's a little bit challenging is that the terminology for the regulatory region is not universal, right? This is a universal drawing of a gene, but people just haven't gotten onto the same terminology, okay? I'll just give you some examples. The molecular biologists are the people who work on it, so is this the right terminology? Well, yeah, we work on it, right, like that, okay? So first, the molecular biology terms, and since we're experts on this, we'll use some of the terms like that. But the molecular biology terms would be, the first one is that what we say is the regulatory region is called cis regulatory units. And I really like to just call it the regulatory region. We got two parts of the gene: transcription unit and regulatory region. The most best name is cis regulatory region, regulatory units, and that's these guys. So a gene is made of two parts: one, the transcription unit; two, the cis regulatory units. And then the cis regulatory units are really made up of two stories: the enhancer and the promoter. Lots of times there are multiple enhancers. We're just focusing on one, but there can be multiple enhancers, and we'll have some examples of that. So things get a little bit complicated, but these are the terms like that. And so in molecular biology, people like to talk about the transcription unit, 6 regulatory units, and they call those cis regulatory units the promoter, is the term they like. And then the best term for this, and we'll talk about it, is response elements. There's a good chance that you've learned about enhancers. Enhancers are super fascinating and famous and important. So the words we just wrote down are kind of the most official terms. Here are the terms that we'll use, and you're welcome to use, right? Like that. What are the terms that we really kind of want to know and learn? And they are the transcription unit, the regulatory region. Sorry, my pen just continued to kind of frustrate me here.
Is that synonymous with
Cis regulatory units, yep. And then enhancer. And that's just because it's used so much across biology, and like all your other professors will want to call it an enhancer. And then promoter. Okay? Now I'm just going to show you, we're not going to talk about it this way, but just to give you an example, in computer science, which is huge because it's used to help discover stuff in DNA and the organization of genes, there's huge amounts of IT work being done in this. In computer science, they call this the gene. They just call the transcription unit the gene. Last time I checked, Wikipedia calls a gene this, like that. And then a computer scientist will call the regulatory region, they'll call that the promoter. See? And you don't have to learn that. I know it's confusing when there are multiple terms. I just want to introduce you or share that reality with you. We're never going to call it a gene and a promoter, because that's wrong. A gene is not just the transcription unit. Of course it's the regulatory region, because you can have mutations that regulate the gene. Gosh, it's just goofy. The reason computer scientists call it that way is because they like to make discoveries in the regulatory region because they're hard to make. It's pretty easy to identify using a computer all the transcription units. And so you say, oh, we found all the genes, but now we're going to make new discoveries like that. So, you know, like that. And I know that's confusing. In microbiology, that's, you know, studying bacteria. The story's the same, but they use very different terms. They talk about operons and operators. The physical story's the same. There are two parts, and they're cis. They're connected. And the two parts are DNA, they're physical, and it's the transcription unit that's going to make the output, and then it's that regulatory region, and they're cis. So whatever the terms are, it boils down to the clay model's the same. And a key concept is that physical nature of it, like that. Okay? So that's a big part of what we're trying to get out of the clay model videos. And the recognition of these terms, you know, those terms are terms about DNA, like that. And they're not changing, right? The transgene unit's not changing and so forth like that, okay? Yep.
So for the, going back to cis mutations, does it have to be like these two things that are associated, connected in this way, like one, like, does it have to be like two parts that are part one and part two in this model? You know what I'm saying?
So probably, maybe. So like it is, so what's going on is like in traditional genetics, the geneticists will find, they'll like screen fruit flies for mutations that have a defect in muscles. And they'll find, there's a fruit fly that's got no muscles. And we're going to maintain it. And it's called a line. It's like a variant, it's like a breed, like that. And they're going to maintain it and study it. It must be mutated in a gene that's important for muscles. And then they look, and here's another one, an independent one, so it's probably mutated in a different location, right? But there's another one, and another one. And so they've got several, and then they want to say, how, you know, these all have defects in muscles, how are these genes related? And so they can do recombination mapping and complementation mapping. And they can map them, and they can say, oh, these mutations are all on the same chromosome close together. And then here's mutations way over there. See, can't see, in traditional genetics, they can't see the genes. They're looking at the phenotype and asking by breeding experiments, what's happening with that muscle phenotype? And so I think you might be asking about this idea that when you're studying, you could have, oh, you know, look, we found a mutation and we mapped it here. It's in the transcription unit. And then in another line, we found a different mutation and we mapped it. It's really close. And later we discover it's actually also in the transcription unit, like that, right? So yeah, you can have, you know, multiple mutations in, you know, you could have a mutation in the enhancer and another time you find one in the promoter, like that, in different animals, but you're mapping them and you're like, wow, these are all cis. They're all really close together. And they do that by recombination to map exactly where along the chromosome is. And then they're like, and then we have just, you know, it's just surprising because we're studying muscle development and we have this one way over here. You know, it's a different chromosome. What's going on with that? And they, you know, they wanted to use terminology for the logic of that. To, you know, we're going to have to explain that someday, but we're just going to call these cis, and cis means physically connected, same chromosome, and pretty close together, like that. And probably one gene. You know, they didn't even know what a gene was. A gene used to be theoretical. It wasn't physical, like that. And then one over here, must be, let's, you know, use a term for that because it's really a funky different thing. We'll call that trans. So that's where that cis regulatory units come from, and it's a nice term because it brings with it the term of the evidence for, you know, the experimental evidence for why you're saying that about the gene organization, like that. Yep. Okay, so, you know, that's what the clay model videos are about. You know, they're plowing into both, oh, we have a situation where we want to talk about a gene, and they're also explaining the organization of a gene and the output of a gene. We're going to have a concept chat. Let me just add one more thing to that. So here's a universal drawing of a gene, and the gene is going to have an output. So there's the output, and that's RNA. For some genes, they code for protein. Like the insulin gene codes for protein. And so it has a secondary output. It's going to be the protein product. So the ribosome is going to synthesize that protein, and the ribosome is going to use that RNA to synthesize the protein. So there's the ribosome getting that job done. Okay? If this gene is like that, then we call this type of RNA mRNA for messenger RNA, because it's literally bringing a message to the ribosome of what amino acids to add. So we're making that. Then, here's another term. For this gene, the general term is it's called a coding gene. And some, but not all genes, are coding genes. Coding genes code for proteins by coding for mRNA. So the output of a coding gene is mRNA, and then next a protein. We can also do those experiments, right? We did experiments to detect RNA. We can also do those experiments to detect protein. In the muscle precursor, we detect no myogenic RNA and no myogenic protein. When we're studying proteins, the most common method is called a Western blot. And we're just trying to detect a protein. Is it there? We're studying that protein. Because, of course, that's a big difference, right? Like that. So that's a coding gene. There are actually lots of genes that don't code for proteins. So here's another gene, and it's called a non-coding gene. Those don't code for mRNA and for protein. They're non-coding genes, and they can code for ribosomal RNA, rRNA, or transfer RNA, tRNA, or small nuclear RNA, like that, or small interfering RNAs, or microRNAs, like that. So we have two different classes of genes. We have one model for gene expression, because all those genes are regulated the same way. They have a transcription unit and a regulatory region that's regulating where, when, and how much of them get made, like that, okay? And so most of the time we're talking about a coding gene, like myogenin, and a coding gene also has another term we want to use, and then we'll have a concept chat, and that is within the transcription unit, a subset of the transcription unit is called the coding region, and that has the codons, the triple letter nucleotides that code for the individual amino acids. Okay? And of course, a non-coding gene does not have a coding region, doesn't code for a protein. All right, so let us have a concept chat. And what this is like is, if you want, you can get up and move around like that. If you want, you can put on a mask. We're thinking about that and being sensitive to that, because we hope, we'll just, over time, we'll do this each time, just go randomly say hi to another person, right? So part of this is saying hi, just a few minutes. And what are 3 important things that you think we talked about today, and chat with a partner. If there's an uneven number, we'll have 3, but we try to have 2. Okay? So go for it. Awesome. Thank you.
Then there were 2 classes. All of the cells have the same genes.
What's that? All right, and so here's the sign-up sheet if you missed that. You always want to do that. And we do this fast. You can say hi and howdy, but you also want to grind through that, 3 important things, and because I'm going to ask 2 people. Or 3. All right, we're just gonna do another minute like that, and just jump through this, and then we'll talk about it.
Possible correlation with, like,
You say
how
a
that.
couple more minutes?
Like one. Yep. Yeah. And well, we'll just go for it. So we can do it like this. So then it's like this. Hannah, what is one thing that you guys talked about that you thought was important from class today?
I think it was really important to know that you have the system trends, how that can affect the product in general.
Yep. So that cis and trans concept, and it's a genetics concept. And something I haven't mentioned is this field of molecular biology is like it was born in the 1950s when Watson and Crick discovered the structure of DNA. And it was born as a result of a marriage between genetics and biochemistry. Like that. Genetics and biochemistry got together, had a baby, and they called it molecular biology. And so much of what we talk about is genetics, like that, cis and trans, and some old school stuff, but it's satisfying, because it's satisfying like with that tractor fixing mentality, physical, right? Physical evidence. What's the data like? Yep. Okay, that's one. Eva, what's one thing you guys talked about that was important?
We talked about transcription regulation and the thus far 3 different parts of why it could be activated or deactivated.
Yeah, where, when, and how much. I mean, you might as well just memorize that because there's not going to be another one. Like there's no advanced course where they say, oh, here's a 4th one. It's where, when, and how much. Like that. Awesome. And then, let's see, I also marked down Samantha. Oh, isn't Samantha here? I thought one of you was Samantha. See, and I'm really sad because in that small town I grew up in, I didn't meet people, right? I'm just like, nobody moved in and out of that town, so I'm not good with names. Joe is here.
Yeah. We talked about how you have the 2 classes of genes, but only one model, and with genes that code for proteins, they're coding genes, they have mRNA as an output along with the protein product, and that they have in the transcription unit, they have the coding region that goes for amino acids.
Yep. Yeah. A week from today, and we're going to talk some more now, so now we have some more time to talk, but just let me mention to follow up on that. A week from today, this Riley's going to come in, and he's going to talk about chimeric antigen receptor, CAR T-cell therapy. And there are really 2 parts. He's going to come in like 4 times during the semester, and there are really 2 parts to that story. One is CAR. CAR is this synthetic gene they build, and they make a synthetic chimeric protein receptor. And a chimera is a Greek word for a monster that's like half human, half horse. That's like chimera. So they're going to build that. So that's part of Riley's story. And the other part is they're going to put the CAR, the modified gene, into a T cell. That's a kind of immune system cell to affect the immune system's activity. And it's going to be a receptor. So it's good. We're going to make a synthetic receptor gene, put it in. It's going to get expressed, right? And it's going to be on the cell surface. And that's going to affect that T cell's immune activity. The receptor is going to bind to the cancer, and the T cell is going to bring in helpers and kill it. Like that. So that's Riley's story, right? And so right now, we know when he talks about the CAR, they're going to be manipulating what? They're going to be manipulating the transcription unit and the coding region, right? They're going to take the coding region for 2 or 3 genes, and they're going to put pieces of them together. And that's going to be the DNA for one new hybrid gene. When you make a new gene and put it into an organism, it's called a transgene. It's a different word, right? Not that trans like we just talked about, but it's just called a transgene. And you make a transgenic organism, right? Like that, or a transgenic cell. And so that transgene is going to be this hybrid gene, and they want to make a different product. They want to make a synthetic receptor product. They can do that by like designing the gene. What part? The coding region, right? And it's just like, oh yeah, duh, kind of like that, duh, you know? And I hate to say that, but you know that happens a lot on a farm where you're like, duh, you know, you got to hook it up, or you need some gas, duh, you know, like that. And I try, you know, like that, you know, because that's just mean. When you're learning it, you're like, duh, you know, like that. But right? That's all in there, all about that, right? Manipulate coding. So, we're setting it up to appreciate the physical nature of it and bring our minds to that, to the clay pieces. On Thursday, Really's going to talk, and then we're going to build the clay model in class, really just like the one in the video, to just kind of bring that physically to you. But then we'll say, we heard just Riley, and we're going to mess and make a synthetic receptor protein by making a synthetic transgene, where we manipulate. You're going to be like, I should take my scissors and cut the coding region, insert stuff in the coding region, right? Like that. It's just super satisfying in that way, like that. I always say, lots of us, we don't know anything about car mechanics. We don't know anything about it. But like everybody knows, if the car doesn't start, you don't check the tires, right? It's that kind of, that kind of knowledge and reality, where you're just like, okay. And all this is like, it's just a lot, right? Like car mechanics, ooh, that's a lot of stuff. When you do open the hood, ooh, there's a lot of stuff. I have a friend who bought land in Denmark, Maine, at the end of a 2-mile freaking driveway that goes up and down, and she has to plow it with a plow truck, right, like that. And then the neighbor's a tractor for backup, and really no mechanical background, right? So it's just a winter freak out like that this winter. It's just like, oh my God. Also, the house isn't finished, so they got a generator, they're working in a camper. All mechanic. You're gonna go off grid, and I'm like, yeah, you are. It's maybe not an electrical grid, but man, you're gonna be stuck out there in nature with a lot of required mechanics like that. So it's like that, and we don't have to be superstars like that, but just so you have that, it's gonna be that kind of talk, right? Okay, so we have a couple more minutes, 1-2 more minutes like that. So let me just show you that we did that, and let me just show you this for a fleeting concept, right? So our muscle videos, right? I picked the muscle because you know something about muscle, right? Like that. Oh yeah, and it's relevant, right? Your muscles can get bigger when you work out, or when you get injured, you can heal, or when you're a child, they get bigger, like that. And that's a physiology story, right? So we're bringing, as our example, a physiological story with that familiarity. But where we're at is really over here in this NO, and there's lots of times you'll know these terms. There are a lot of muscle proteins. They're specific to muscles. Myogenin is binding to those genes and activating them, right? You gotta have them. So we need to make those proteins. They're not present in the precursor. They're present in the muscle. Their RNA is present in the muscle. It's not present in the precursor, right? So it's a good example. It's an accessible example. The story is over here in this development way. And lots of stories are because during development, there's a lot of gene expression changing, right? We're making stuff, making a brain, right? We've got the genes. We've always had them in here for our parents. Now we're going to make them. In our lab class, we're going to study insect metamorphosis a lot because, well, you already made a caterpillar. Now you're going to make a moth. How are you going to do that? We're going to activate all those moth genes like that. Yeah? So it's like that, right? So that's where we're at. And it's really fascinating because when a person works out and they build muscles, those satellite stem cells get activated to make more muscle cells. So we do know the details of the cellular processes that lead to the physiological changes like that. Or when an animal migrates, birds migrate, they build up their muscles like that. And they have, we understand the mechanism. So it's like that. It's an accessible example, but we're really going to call it clay model videos because of what I talked about almost all today, and that's this universal example. Okay? So we're good to go. If you